Here we go again. This is question 2 on the mock paper. So, photograph shows all the chromosomes. Oh, this looks more like component 2, doesn't it? That's great. Photograph shows all the chromosomes from a blood cell of a mammal. And already I'm looking at this and I'm thinking, those two are matching. Those two are matching. These two, not so much. So, already, because we've got pairs, this is 2N diploid, and I'm thinking that that's a Y chromosome and that's an X chromosome because they haven't got a pair. So, let's see what they're asking. Explain why red blood cells could not have been used to produce the photograph of the chromosomes. Red blood cells, again, bit of synoptic, have no nucleus. That should be straight at the top of your head. Use the photograph to deduce the sex of the mammal and explain your choice. Male, as it has X and Y chromosomes. Ooh, in this mammal, how many chromosomes will be present in a kidney cell? So I'm thinking kidney cell, they're going to be in pairs, that'll be 2N, that'll be N. These are in pairs, 1, 2, 3, 4, 5, 6. 6. Gametes, half the number, 3. And oh, our favourite sort of question. Draw something. The circle represents the outline of the mammal cell at metaphase 1. The dotted line, the equator. Complete the drawing to show the spindle. That's my spindle. That's my centrioles there. I'll put 2 spindles on for fun. Show the spindle, and do we have to label anything? Oh no, do I have to label anything? That's a miracle. I might do that anyway. And show how the sex chromosomes. So the sex chromosomes. We've got one that's kind of long, and I'm going to colour it in a bit, make it a bit wider so it looks a bit more like what I've got in front of me. And then the other one is a little triangle one. That'll be on the other side of the equator. So, I want some labels on. Get my trusty ruler out. Because if I label it, the examiner knows what they're looking at. X chromosome. And they know that I know what I've drawn. Y chromosome. Usually, when you've got to draw something, there'd usually then be marks for labels rather than for the quality of your drawing. Okay, starfish reproduce sexually by spawning. I'm not even bothering reading its proper name. It means red star, bizarrely. When starfish spawn, the males release spermatozoa and the females release oocytes in large numbers. The oocytes are suspended at early prophase of the first meiotic division. The nuclei of early oocytes are called germinal vesicles. Lovely. Breakdown of the germinal vesicle indicates the resumption of meiosis reinitiation. Reinitiation is naturally stimulated by contact with spermatozoa. Scientists have shown it is possible to stimulate reinitiation by exposing the oocytes to suitable concentrations of calcium ion, so presumably instead of sperm. Seawater has a typical calcium ion concentration of 14 milligrams per decimeter cubed. I don't quite know why they've told me that yet. Suggest a suitable hypothesis for this explanation. So let's do that first. Hypothesis. What do we think? So what we're trying to show is that calcium ions will reinitiate meiosis. So we want a relationship between them for a hypothesis. So the greater the concentration of calcium ions, the more, what should we call it, reinitiation of meiosis will happen, perhaps. Might just leave it at that. More the reinitiation, I can't even say it, reinitiation of meiosis. I think if you wanted to write a null hypothesis for that, you would have to say the concentration of calcium ions has no effect on the reinitiation of meiosis. And I don't think it matters. It says a suitable hypothesis, so I think you could go either way on that. So that's that done. Tick that off. Design an experiment to investigate how calcium ion concentration affects meiosis in starfish. So I'm going to need to do IV, DV, controlled variables. That's just a kind of given really, and I want 2 of them. What does it go on to say? Make sure you suggest suitable values for the independent variables. So we need values in there as well, and that's why they've told us that. As well as identifying the dependent variable and 2 control variables. Oh, it's told us what to do. Well, always, but it has on this case. So, the independent variable is concentration of calcium ions, and we're going to use, so we want values that are around 400, maybe a couple lower, 400 and a couple higher. So I'm going to go for 300, 350, 400, 450 and 500 milligrams per decimetre cubed. Don't forget to put units in. So that's the independent variable. We've done the values. Dependent variable is the number, that's all you can do really, is look at them down a microscope and count them. Number of oocytes where meiosis is reinitiated, and you could actually put down how they're going to measure that. So, you know, how many of them where the germinal vesicle's broken down, maybe. So that's the dependent variable. Now we need 2 control variables. So the one that I would always recommend that you put down is temperature, if it's appropriate. So if we're not altering temperature, and we're not. So controlled variables. And again, don't abbreviate this. If you're going to say temperature, don't write temp. Temperature of the solution in degrees centigrade. And, because we need 2, so you could have how long they're left in the solution, maybe. You could have the same starfish. And let's do that. Taking oocytes, because they'll release billions, I imagine, from, so if they're releasing into the sea, definitely big numbers. same starfish. And again, I could have put same concentration of anything else in the medium, the pH, time that you've left them. It doesn't really, as long as you've got two sensible things. So that's for 6 marks. So I'm thinking 1 for the hypothesis, tick. Suitable values, independent variable, dependent variable, and 2 controlled variables. Job's a good one. Okay, some human fertility problems are due to poor morphology, abnormal shape, or poor motility, not moving normally, of spermatozoa. These causes of infertility can be treated by a special type of IVF called intracytoplasmic sperm injection. That sounds exciting. It differs from conventional IVF in that a single spermatozoon is injected directly into a secondary oocyte, and they're showing it here, instead of it taking place in a dish and just flooding the secondary oocyte with loads of sperm. However, fertilisation does not always proceed. A solution of calcium ionophore may be used to stimulate gated calcium ion channels in the oocyte plasma membrane. Ionophores are molecules that facilitate ion passage in or out of cell membranes. Use your knowledge of the structure of the plasma membrane. So here we are, we're on synoptic again. To state what's meant by the term gated calcium ion channel. So, a calcium ion channel, is a channel made of protein. It could be an intrinsic protein. Try and get as many terms as you can remember. Made of an intrinsic protein and gated means it can be opened or closed. In humans, before fertilisation, the oocytes are suspended in metaphase 2. You know that. Explain how the ionophore molecules might improve the success rate. So what did they say was causing it not to be? They told us what it was, but said that fertilisation does not always proceed to completion. So presumably, I'm thinking there, meiosis 2 doesn't reinitiate. So this is a beautifully scaffolded question. So they've talked about calcium ions reinitiating meiosis on this page, and on this page they're saying, well, how could you make it happen using this technique, these calcium ions? So ionophore molecules they facilitate the passage in and out of the cell membrane of the calcium ions. So ionophores would let calcium ions into the cell and stimulate reinitiation. How do I know I need to talk about that? Well, apart from the whole question is because it tells you that they're suspended in metaphase 2, so you've got to reinitiate that and make it go to completion. And if you remember then from doing fertilisation, you'll make pronuclei, and it's those two that confuse together. There we have another one. Still can't remember how to switch it off.